So we suggest to continue with the last session today. And our first speaker is Michał Świtnicki from Opus University.
Okay, thank you. Thanks to Pavel for inviting me. It's been a great conference so far, and it surpassed my expectations in terms of the content and the relevance to my work also. So today I will speak about my integrative method for analysis of cancer that improves identification and classification of perturbed genes. I realized that I don't need this really because it has been popping up. But just to remind you, cancer is a disease characterized by uncontrolled cell growth and proliferation. For cancer to develop, genes regulating cell growth and differentiation must be altered. And these mutations are then maintained through the subsequent cell divisions and are thus present in all cancerous cells. Particularly important from cancer's perspective are oncogenes and tumor suppressor genes. Gene expression profiling is a technique used in molecular biology to query the expression of thousands of genes simultaneously, and in the context of cancer, scientists typically focus on the aforementioned oncogenes and tumor suppressor genes. And the paradigm says oncogenes are upregulated in cancer and kept low in the healthy tissue, and it is opposite for the tumor suppressor genes that are normally highly expressed in healthy tissue and lowly expressed in cancer. In the past, gene expression profiling has been used to more accurately classify tumors, and the information derived from gene expression profiling often has an impact on the predicting of the patient's clinical outcome. Here's just some signature for prognostication. So methylation of cytosines in CPJ sites, often referred to as DNA methylation, is a common epigenetic signaling tool that cells use to lock genes in the off position. In recent decades, scientists have learned a great deal of information about DNA methylation, including how it occurs and where it occurs. They have also discovered that methylation is an important component in numerous cellular processes, including embryonic development, genomic imprinting, X chromosome inactivation, and preservation of chromosome stability. Given the many processes in which methylation plays a part, it is perhaps not surprising that scientists have also linked errors in methylation to a variety of devastating consequences, including several human diseases like, for instance, cancer. And here's one of the mechanisms by which cancer is given rise by methylation, by changes in methylation. And just to show you an example of the clinical use of methylation, here's a recently published paper from our institute that uses low and high methylation signature in this region to predict or to split the cohort into high survival and low survival of the prostatectomy patients. So there are many ways that gene expression is regulated in eukaryotes, but DNA methylation is one of them. And in terms of explanatory power of DNA methylation for changes in expression, and here I'm showing some real data from cancer. The greens are the healthy tissue data, and the reds are the cancerous samples. And what I'm showing here is the expression on the left, and sorry, on the y-axis, and on the x-axis is the promoter methylation and gene body methylation. And the reason I split it is I will come back to that later, just in a second. So So the traditional paradigm only considered the promoter methylation of CpG sites, and its high levels were linked to the suppression of the genes where it was occurring, thanks to the insulatory power of properties, and thus they were preventing, this high methylation was preventing from binding of transcription factors. Some recent advances, however, now also consider a different mechanism of influencing gene expression by methylation, and models exist showing that DNA methylation in the bodies can also affect transcript abundance by promoting its elongation during the RNA polymerization, and therefore increase gene expression. So the paradigm, the expected behavior is negative correlation of promoter methylation with expression and positive correlation of gene body methylation with expression. So now looking further into the real data set of breast cancer adenocarcinomas from the TCGA data, from the TCGA cohort consortium, we can see the transcriptome-wide distributions of correlations between the gene body and expression and promoter methylation and expression. And we can see that there are both enrichments of, say, paradigm-like interactions and also counter-paradigm interactions. So there are various mechanisms, and it's hard to generalize. What's more, when we consider the differences between healthy and tumor groups in terms of DNA methylation to gene expression interactions globally, we can see that there's also an enrichment of changes in the degree and the directionality of the correlation. And this known distributions is what you would expect by random. And the color distributions is what we actually see. Therefore, it would be highly beneficial to analyze both data types jointly, allowing for simultaneous assessment, including the interplay between the DNA methylation and gene expression. And this would then allow us to very accurately evaluate individual samples. Let's now consider this small cartoon. Which sample is more likely to be observed? If the relationship, the natural relationship is like this, which one is more likely? Is it this one or that one? And clearly it's the less likely to be observed is this one, which has low expression and relatively low promoter methylation. Okay, so we have assigned the primary purpose for our method to enable the assessment of the combined genetic and epigenetic alterations of individual genes in cancer with respect to the control behaviors in the healthy samples. An important and highly desired feature of the method is an ability to assess the perturbations with regards to the interdependence among the model variables, if present. In traditional analysis, data is often analyzed separately, and independence is normally assumed when combining individual results, for example using Fisher's method, which is, as I have just shown you, is often not the case. We'd like to address that to improve detection of the perturbations in cancer, and also to enhance, to better understand cancer's biology, to identify candidates, and to better discriminate healthy or benign samples from malignancies. To demonstrate the proof of principle, we analyze gene expression with respect to DNA methylation in a rich clinical data set of 75 tumor paired normal samples obtained from the Cancer Genome Atlas Consortium. And we use, for expression profiling, we use the RNA-seq from the cohort that give us transcript abundances of over 20,500 genes. And for DNA methylation, we utilize the 450K methylation microarray platform from Illumina that interrogates over 480,000 CpG sites throughout the genome. We further divide the methylation probes into promoter and gene body associated CpG sites. And the suitable overlap is 17,728 genes that we have both expression, promoter methylation, and gene body methylation. So in an attempt to address these issues that I have just raised, we make use of the probabilistic graphical models. And probabilistic models are a marriage between probability theory and graph theory. Here's some probability theory. And they provide a natural tool for dealing with two types of problems, uncertainty and complexity. Fundamental to the idea of a graphical model is a notion of modularity. And a complex system is built by combining simpler parts, ensuring that the system as a whole is consistent, and provides a way to interface models to the data. And the graph theoretic side of graphical models provide both an intuitively appealing and interface by which humans can model the highly interacting sets of variables, as well as a data structure that lends itself naturally to the design of the efficient and general purpose algorithms. Let's now focus on the model. It's a gene-centric approach that takes into account all the available CpG sites, and the three modules, the three individual modules, the promoter methylation, the gene body methylation, and expression modules, evaluate individual data types, where we model the likelihoods. It's a probabilistic model, so we model the likelihood, the probability distributions in each of these, on each of these nodes. And we model the technical variance in the expression model using the Poisson distribution and normal distributions for the methylation models. The full model, however, further models the relationship of the expression with promoter methylation and gene body methylation. So now we have combined the individual parts. We have two uses of our model. First is a genome-wide differential analysis. In such scenario, we compute the D statistics using the tumor-only and healthy-only models. So we built three models actually, one for the tumors only, another for healthy only, and then we built a combined model. And the D statistics tells us how much of a benefit we get by treating them separately, the tumors from the normals. This is the same test statistic that is calculated in the likelihood ratio test. And then we calculate the random distribution of the D statistics per gene and calculate the Z-scores for genome-wide ranking of genes, or to perform actual evaluation. And the second scenario is the classification of the new sample by the means of calculating a posterior probability for each sample at hand, using the tumor and tumor-specific and healthy-specific models. And this probability tells us how likely it is that a given sample is a tumor, or is a tumor. If we analyze the breast cancer data set using the individual submodels and compare it with some state-of-the-art methods, we can see the concordance of the results. The least degree of agreement is seen for methylation models, because the traditional analysis of methylation, of region-wise methylation, combines the individual measurements from probes into a single matrix, say a mean, and then for each sample this is taken into analysis. And in our approach we model the entire landscape of the methylation. We take every CpG site into account, And this allows us, this in turn allows us to obtain better idea of the complexity of the DNA methylation landscape and to account for the heterogeneity within the cohort. And we compare against a tool called HR, and we also compare against a method called IMA for the methylation. Now it's a good time to discuss the heterogeneity of cancer cohorts with respect to the normal behavior as observed in the healthy tissue. Let's look at some examples from the breast cancer dataset. These are some genes. And it's again the relationship between expression and promoter methylation and expression versus gene coding methylation. So from these plots, we can see that typically there's one state that characterizes the healthy state, the healthy tissue very well within a certain biological margin. And tumors, however, can exhibit healthy-like or perturbed behavior. What's more, within the margin of perturbed behavior, we may see different modes. The meaning of such modes probably reflects the different molecular cancer subtypes, which we can hopefully elucidate using the integrative methods. We may summarize such plots across the entire genome in the breast cancer cohort by looking at the empirical distribution of the variance ratios of the tumors to variance in normals. And again, this null distribution is what is expected by random, and we can see that there is significant enrichment of high standard deviation ratios. So this tells us that it's a significant effect in the dataset. And to exemplify our model's superiority in terms of detecting genes with multimodal nature, with high variance and in the tumor dataset, in the tumor group, we simulate a dataset stratified by a fraction of the true tumors in the tumors group, and from these plots show results of from our models and also from the state-of-the-art corresponding method. And we can see that our methods are not only more can detect more signal, but they also retain more signal throughout the series, which shows that we are resistant to this effect, unlike the state-of-the-art methods. If we now look at the full model performance in comparison with the traditional approaches of combining the data, meaning the Fisher's method, we can see that performance is quite comparable in the given data set. But this data set contains a relatively pure signal, and meaning that the data that was here contains only single mode. Our individual models, as well as the full model, again retain more power as we progress through the series. And now, if we simulate a data set stratified by the delta correlation, the difference in the correlations, degree of correlations in the tumors and in the healthy tissue, and on top of that, we decrease the signal to noise ratio, we can see that now the joint model actually starts to benefit a lot over the individual approaches, even our probabilistic graphical models for a single data type. And as we increase the signal of the change in the correlations, we can achieve very good performance, and it's a lot better than individual analysis combined. if we now look at the global comparison of ranks produced by our full model and combinations of p-values from state-of-the-art methods by the Fisher's method, we can see a general agreement, and the Spearman's correlation coefficient is 0.725. And I had some gene sets highlighted here, but I'm running out of time, so I will just skip them. So looking at the top perturbed genes, according to our method, we can see that discriminating healthy tissue from tumors is not too difficult, as illustrated by these plots. Our method generally agrees on the many of the candidates, for instance here. However, we find some that would be typically missed by investigators, like NECAB1. And NECAB1 is a cosmic gene, meaning that it's mutated in various cancers, and it was found often mutated in prostate, in cancer, lung cancer, endometrial, ovary, large intestine, and also in the breast cancer. Okay, it's a quick table. We can predict sample labels in an independent set of 705 tumors and 81 healthy tissues from the same TCGA cohort, but that was not taken into analysis. And by combination of the multiple genes, we can achieve remarkable performance. And if we look at this NECAB1 gene and its performance by the, say, multiple logistic regression combined variables, we can see that our model is performing much better by taking this correlation or interdependence into account. And moving on to a more difficult task of predicting the tumor aggressiveness within the TCGA breast cancer cohort. I selected 14 tumors that progressed after initial treatment within the first 1,060 days, and 57 tumors that did not progress within this period, after this period. And such a small cohort is quite problematic, since we can't afford to have an independent prediction or validation dataset. And the volume of the data to learn the multi-dimensional distributions comprehensively is not adequate. Nevertheless, here is a list of the top 10 genes found by our method. And looking at the underlying data, we can see that the problem is difficult of discriminating these distributions. The red is the progressed and the blue is non-progressed. However, the top candidates like SERPINE3 and ZNF706 are quite credible for their roles in cancer progression. For example, SERPINs, the largest family of endogenous protease inhibitors, are involved in various pathological processes, including cancer progression. Here we detect the SERPINE3, one of the representative of the SERPIN family. And the top two is the ZNF706 gene, which was recently used in a gene expression panel for prostate cancer progression. I'm not sure if I have time to. Oh, okay, that's fine. It's fine. We also compared our method against using expression alone for this progression task, because a recent publication in the scientific reports said that it's actually the gene expression that is the most informative modality, and it significantly outperforms other modalities, and also